Thank you for joining the NAA today for the State Natural Areas Program Roundtable, Rare Plant Propagation and Reintroduction, Questions and Considerations, featuring two speakers, Matthew Albrecht and Elizabeth Olson. But before we get to our topic today, I'd like to just tell you a little bit about the Natural Areas Association. The Natural Areas Association serves those dedicated to the management and restoration of ecologically significant landscapes in North America. Protecting nature requires reliable science to inform practices on the ground, and a network of stewards who work tirelessly to protect and manage and restore land and water biodiversity. We are a community of peers and colleagues who become a resource that can respond to questions, propose ideas, share methodologies, and advance best practices. For over 40 years, NAA has supported your work in natural areas management by bringing you the latest science through things such as the Natural Areas Journal, which is a quarterly peer-reviewed journal, and the January 2022 issue is now available. We also host the Natural Areas Conference. Our call for proposals is open and the deadline is March 1st, but we hope you won't wait that long. 2022 Natural Areas Conference will feature plenary workshops, breakout sessions, hands-on workshops, field workshops, posters, and more. It's being held in the beautiful Duluth, Minnesota, and will feature on the wide and complex variety of natural areas there. We are also thankful to Esri for sponsoring this webinar, Geographic Approach for Natural Areas Management, to make it available at no cost for all. In May, our webinar will be Hopes for the Hemlocks, HWA Management Approaches on Public and Private Lands in North Carolina. And in June, we will focus on fire ecology in the upper Midwest, effects of the fire seasonality and intensity on controlling resprouting woody plants. I'd also like to tell you about our Stewardship in Action virtual field workshop, Sustainable Summits, Managing Public Access for the Protection of Rare Plant Communities. This event was filmed on site in Southern Virginia at the base of the Appalachian Mountains, and then was presented virtually in January. It is a full, compact 2-hour workshop that deals with a lot of the issues of managing public access in our spaces while protecting the rare vegetation there. And now I'd like to talk to you about today's program. So I'd like to thank you for joining us today. NAA hosts 4 roundtables a year. 3 are virtual like this one. And hopefully in November, fingers crossed, we're moving past COVID, we'll be holding in-person roundtables during the September Natural Areas Conference. The virtual SNAP Roundtable schedule is May 12th and November 17th, which will be virtual. And then in September, the in-person state reports. SNAP Roundtables are practical and focus on the needs of and interests of state natural areas program managers. If you have a topic you'd like to suggest for a future roundtable, we welcome your input. Now let's turn our attention to today's program, Rare Plant Propagation and Reintroduction. This is a topic that we are all dealing with, and it is more complicated than we realize. To that end, I'm delighted to welcome 2 speakers today whose background and experience in this area will not only inform our practice, but will better prepare us to explain the complexities related to the preservation and relocation of rare plants. So let's get started. Our agenda today will feature 2 talks, Rare Plant Propagation and Reintroduction, Questions and Considerations for Natural and Historic Resources Lands in New Jersey, and then Restoring Populations of the Federally Endangered Pines ground-plum using Adaptive Management. We will also include a couple stewardship shorts and then have open discussion. Our first speaker today is Elizabeth Olson. Elizabeth works for the New Jersey Department of Environmental Protection in the Office of Natural Lands Management, where she assists with field surveys for rare plants and researches the ecology and management of state-listed rare plant species. Liz is also an adjunct instructor at Stockton University. She studied ecology and environmental sciences, earning a BS from Temple University and an MS from the University of Maine. Our second speaker will be Matthew Albrecht, Ph.D. He is a conservation scientist at the Missouri Botanical Garden and Center for Conservation and Sustainable Development. His research interests include the restoration of ecology of rare species and degraded habitats, understanding the drivers of rare plant population dynamics and the implication of seed traits for conservation and restoration. He works closely with governmental agencies to develop management solutions for threatened plant species. He's also authored or co-authored several dozen articles and book chapters, including the Center for Plant Conservation's Best Practice Reintroduction. And now let's hear from Liz.
Because there are a lot of terms that are used to refer to this practice of outplanting propagules of rare species to restore populations. So you'll see some projects refer to an introduction, an augmentation, a relocation, a transplantation. And all of these terms, they do have very specific meanings and are used, in specific context. But just for the sake of simplicity, here in this presentation and also in the written report, I'm just going to stick with one term, reintroduction, and I'm going to use that as very much a broad blanket term to encompass all of these types of outplantings of rare plants in restoration projects. So I used a wide variety of sources, including a lot of overview papers and meta-analyses. There are also a lot of case studies and experiments that were more focused on single species reintroduction attempts. Also, I looked at a lot of policy and guidelines and websites from state governments, federal governments, and NGOs and other organizations who are weighing in on this topic. And so I have a lot of references. It certainly wasn't a complete exhaustive search of the literature, but it was pretty thorough. And so the goals of reintroduction projects have some very ambitious goals, which is great. And so the goals are to establish resilient and self-sustaining populations that have the appropriate amount of genetic diversity that's needed to adapt to a changing environment, including climate change, and ultimately to prevent these rare plant species from going globally extinct or being locally extirpated, which is, can be just as important. And these practices of plant propagation and reintroduction have really advanced, especially in recent decades. There's absolutely some wonderful success stories. And certainly for some species that are on the brink of extirpation or extinction, a reintroduction project really might be the only option left to save it. So reintroductions are certainly a very important tool for the recovery of rare species. However, there are also some negative implications, some potential unintended consequences that a lot of folks have concerns about when conducting reintroductions. So first, it doesn't necessarily address the root cause of species decline, which is often habitat degradation or destruction. So there is a potential for too many resources to be diverted away from good habitat management, which really needs to be the ultimate priority. And also, reintroduction projects can be used to rationalize the destruction of natural habitat. This sounds like a horrible, how could this ever happen? It actually has happened and it's been documented. One example was given in Falcon Olwell, where in Australia, there was a very rare orchid, and researchers worked for a long time trying to develop ways to propagate this rare orchid. And they finally did, they succeeded, but then that knowledge was used to then justify the destruction of that orchid's, one of its last remaining remnant sites. And so, terribly, that actually does happen. And other unintended consequences, creating new plant populations, it can obscure the natural range and ecological patterns of plant species if those newly created populations aren't known to the community. And this also has occurred. It's been documented for these two species that I have shown on the screen here, the Plymouth gentian and the hard shield fern. They're both rare plant species. And in both cases, researchers were, thought they had discovered new occurrences that were outside of the known distribution or range that was known for these species. But in both cases, it turned out that that new occurrence was actually a planting, an undocumented planting. So there was a lot of confusion and wasted time and effort, all because of a lack of communication with the natural resources professionals and with the natural heritage groups that work in that region. And this is something that I make a point of kind of delving into a bit more in the report, which is just the importance of collaboration among experts and natural heritage offices, because really the most successful projects are the ones where you have a multitude of folks with professionals with different expertise coming together, working together on the project, communicating with the natural heritage offices. And it's really that collaboration and communication that's so key and important. And finally, there can be implications with the state or federal laws regarding rare species, because if introductions increase the number of populations, then a species could potentially and very abruptly lose any legal protections that it might have had, just based on the fact that there are more populations, when in fact, and a rare species could potentially be prematurely downlisted based on these new created populations, but actually the long-term survival of those new populations is very tenuous, very uncertain. So none of these reasons listed here they're not actually reasons to not attempt a reintroduction, but they're just all issues that have been expressed by experts as a reason to proceed with caution. We really need to consider how reintroduction work is heard and interpreted by others like developers or legislators who might take the news of some short-term success and twist that into the belief that we've got this all figured out, and moving plants or translocating plants is easy, and just the twisted or the hope that that we've got it all figured out, and then actually protecting natural habitats is not really that important. And we know that's just not the case. And so in addition to those previous potential unintended consequences, there are also some very concrete risks and challenges with reintroduction projects. So first, there can be harm to the source population and site, because the process of collecting rare plants can be harmful to the source population and the source site. And what I mean by that is the population and the site that you're getting your plant propagules or plant materials from to do the reintroduction. Removing any plant parts can lead to future reductions in its abundance, or it can reduce the genetic variability of that population. There can also be damage to the recipient site, meaning the outplanting site. And the outplanting site and the source site are sometimes the same, sometimes different. But regardless, the whole process can potentially be damaging to the outplanting site. It's a risk that we take. It's a challenge to overcome. There could be trampling, soil disturbance. We could possibly accidentally be introducing invasive plant propagules or plant pathogens unintentionally that could just be transplanted to the new site or transported to the new site during the outplanting process. And there also could be a loss of genetic diversity or fitness in the process, because at each and every stage in a reintroduction project, there is the potential to lose genetic diversity or to negatively shift the genetic structure of the population, so that moving forward, it might not be so well adapted to the site. And another challenge is that often not a lot is known about many of these rare species and their habitats, and specifically their specific microhabitat needs. And actually, this is not understanding the specific microhabitat requirements is one of the main reasons why reintroductions don't succeed. And so this is a figure from a meta-analysis of reintroduction projects, and it shows the reported reasons why various reintroduction projects had failed. And you can see a lot of these reasons really boil down to lacking a full understanding of the habitat requirements and the species biology. And I also want to draw your attention to the tallest bar here. About a third of the reported reintroduction projects. For about a third of them, the reason for failure was completely unknown. So that's just something to think about, something to keep in mind. It often might not even be obvious why it failed. So getting back to risks and challenges, these types of projects are a very long time commitment. It can take many, many years of planning and preparation just to lay the groundwork for a solid project. And then the outplanted populations might take many years to fully establish, might take a decade to fully stabilize after establishment. And so the plants and the whole site are going to need regular long-term maintenance. And even still, with all that effort, all that long time commitment, that your success is far from guaranteed. So definitely challenges. And finally, reintroduction projects can be quite expensive. They often will require some type of funding for each and every step in the process. So knowing all of these risks and challenges, there are still plenty of cases where a reintroduction project is absolutely necessary for the recovery of a species. So first we need to ask, is the reintroduction project justified or not? And the Center for Plant Conservation has excellent guidelines for this, for determining whether a proposed reintroduction should actually proceed or not. So this is straight from their 2019 publication, and they advise that a reintroduction is not justified if it's going to undermine the imperative to protect existing sites. So as I mentioned before, the priority needs to be habitat protection. So if a reintroduction is proposed as an excuse to not conserve and protect habitat, then it should not proceed. And if existing threats have not been minimized or managed, then reintroducing a rare plant is not recommended because it's still going to be under those threats that it's facing now. And likewise, if suitable habitat is not available, or if it's not understood, then the risk of the project failure is pretty high. And if the source population can't sustain the removal of individuals or propagules, then it's not going to be worth the risk of permanent damage to the source populations. And finally, if the species has not been thoroughly researched, so basically they're saying that you do need to have a solid understanding of the species biology and ecology before attempting a reintroduction. And this is not to say that everything has to be understood completely fully, but as best as possible, solid understanding before moving forward with a reintroduction project. All right, so in all of these cases, a reintroduction is not justified. But there are absolutely many situations where it does make sense to attempt a reintroduction. And again, the Center for Plant Conservation has advice on this. And they advise that if all of the following conditions are met, then it's justified. So if the distribution of the species is known and the populations are in decline, and that is a very important point. There are some rare species that are just naturally rare. Their populations may be very healthy and not in decline. It's just a naturally rare plant, and that's fine. We want to make sure resources are being put towards those species that are rare and are declining. And also if the in situ management options are inadequate for the long-term conservation of the species. And and also if the threats have been identified, the threats are known, and there's high risk of extinction or local extirpation. So they say that if all of these conditions are met, then a reintroduction may be justified and you can go ahead and proceed with caution. So there are many elements that need to be addressed in planning a reintroduction project. Our report goes into detail about all the various topics that should be addressed. I'll just kind of run through them briefly here. But I first want to stress that the report that I wrote is absolutely not intended to be a how-to guide for rare plant reintroductions. That's just way beyond the scope of the report. But we do provide a lot of resources for folks to, to look for that kind of assistance for how to go about this type of project. But so here are some of the important elements of a well-designed reintroduction project. So first, before even beginning to plan to implement the reintroduction, it's important to have a very thorough understanding of the species, and that's going to include its conservation status and why exactly is the species rare, what are the threats that it's facing. For example, this could be habitat loss, it could be invasive species encroachment or a legacy of fire suppression, a lot of potential threats. And we need to know the details of the biology and ecology of the species. So this could be everything from its germination requirements, its, its seed production, its dispersal mechanisms, etc. And also understanding the species genetics is important too, since a primary goal for restored rare plant populations is that it's for them to be adapted to changing environmental conditions and resilient to environmental change. So it's important to know if those remnant populations have loss in genetic diversity or not, what it really is going on with them genetically. And here I wanted to mention the case of the Tennessee purple coneflower. It's endemic to this very small region in central Tennessee. And the recovery and reintroduction research included genetic testing, and they were able to find that there was very little genetic diversity among these subpopulations. And so the Tennessee coneflowers, different subpopulations were all very genetically similar. So from that, they knew that they could combine seed from the different subpopulations, for this reintroduction effort, and it wouldn't result in genetic depression. But you can imagine that this would be a very different case for a species that's also rare, but that has a lot of a wider distribution and more disjunct subpopulations. A species like this, Hearst's panic grass, has a very scattered distribution and would more likely have very locally adapted genotypes. And so it could suffer from genetic depression. For example, if seeds from New Jersey were used for a reintroduction project down in Georgia, they might not be locally adapted, and it probably wouldn't be advisable. So important to understand the genetic structure of the species you're working with, and how can you, appropriately support that. And finally, how to propagate the species is really important to have a handle on before investing too much into the project. Some species are just extremely hard to propagate or to break that seed dormancy. And one example is this species here on the screen, Ruth's golden aster. In a reintroduction project, researchers were finally able to figure out how to break seed dormancy. It required first drying the seeds, then treating them with alcohol and fire and bleach, and then fresh water, and then finally being able to put those seeds into a soil medium, and then they would germinate. So it can get very, very complicated. Moving on to the implementation stage of a reintroduction project. First, projects should really begin with a good experimental design. so that the results will be best, so that you'll be able to kind of pinpoint which methods led to success or failure and why that was the case. Very important to set that up correctly. Also, all projects need to find optimal sites for outplanting, and this is not an easy thing to do, but you really need to make sure you have identified suitable habitat, meeting the habitat requirements of your target species, because as I said earlier, the inappropriate habitat is a primary cause of reintroduction failures. And I wanted to share this one interesting example. This species on the screen here is Harperella. It is a federally endangered species found in the southeastern US. And a reintroduction project found it has a very precise microhabitat needs. It grows on streamside cobble bars where it needs at least 6 hours of full sunlight every day. And it needs some low level of flooding disturbance during part of its life cycle, but it can't tolerate heavy, intense flooding. And so you can imagine this is, it's just very precise combination of habitat requirement needs, and it was difficult to find those outplanting sites. And if the target species is dependent upon other species, those other species need to be present at the outplanting site for success. And a great example of this is this species, salt marsh birdsbeak. This was reintroduced successfully in California, but it has 2 co-species that it's dependent on. So first, it has a preferred pollinator, which are solitary burrowing bees. So if those weren't on site, then the outplanting would fail. And secondly, this is a plant that's a hemiparasite, and so it needed to be planted close to native specific native perennial plant roots that it could parasitize in order for it to survive. So next, in implementation, there are a lot of details with the outplanting process that are important to consider. Details about the amount and the density and the spatial pattern of the outplanting can be very important to survival and success. Some plant species are going to do better if they're planted as clumps. Others need more space in between the individuals, room to grow. So all these little details are really important to consider. And also whether to use seed or seedlings can be extremely important to the success of the project. And long-term monitoring is essential to reintroduction projects, because any early successes, there might be early successes as in 3 years you have survival, 5 years you have reproduction, but those early successes absolutely don't guarantee you long-term success in a reintroduction project. So long-term monitoring is essential. And researchers really need to have a strategy for long-term monitoring. This is going to enable them to catch any signs of decline before it's too late. So for example, you might have planted in a drought year, and so your plants would need a lot of supplemental watering so that they can, get established and make it through that drought year. Or they might need protection from herbivores before they fully, while they're young and tender, before they fully establish. And also going back again to the example of salt marsh birdsbeak, I just found the publication on the species so thorough and so interesting. So another example here, it was because of very long-term monitoring, very dedicated, decades-long field work that researchers found that this species has the reason why it has these dramatic boom and bust cycles. And so they were noticing that some years there'd be thousands upon thousands of individuals. A few years later, there'd be fewer than 100. Very disconcerting, very troubling, but because they stuck with it and were diligent with the monitoring and data collection, they found that there were, there were reasons for the boom and bust cycle, and it was kind of a natural system. And so this species' population cycles were dependent upon things like temperature and rainfall and salinity in the salt marshes, and also a 18-year lunar event cycle that affected the tides that then affected salt marsh birds' beak. So 18-year cycle, it could only be through long-term monitoring that they would have found this out. Okay, and through long-term monitoring, you'll be more tuned into the site management needs as well. So the whole outplanting site itself is probably going to need some ongoing management to continue to provide the best habitat conditions. So the site might need management actions like prescribed fire or invasive species management or canopy thinning, a variety of things. So it's again, long-term time commitment. And there are complications that are going to arise because of the nature of a long-term project. And continued funding is one issue, staff turnover is another. I mean, these are kind of inevitable in long-term projects of any type, but you know, they just need to be planned for and, you know, expected and planned for the project's success. We don't really want to think about things like that happening, but if we plan for it, you know, it works out. All right, so finally, we include disseminating or reporting on results as a very important project component, because there is this strong bias in the scientific literature to just publish on positive results and positive outcomes, but really we need full reporting to understand, you know, where were there successes and failures and unexpected events. You know, why did projects work? Why didn't they work? It's equally important information. And so if we can be sure to get the word out on our projects, no matter what the outcome was, it just helps the community as a whole to learn and get better and improve. Even if a project has a setback or a failure, it does provide useful ecological information. So that was just a brief summary of all of the topics or components that a reintroduction project needs to take into consideration. There are many, many more details in the report. And we end the report with a list of resources for folks to consult for more information and guidance. And I do hope that you'll take the time to download this report, bookmark it, save it, share it, refer to it as a reference. One of the resources included is this checklist for those who may want to design a reintroduction project or who may need to review reintroduction proposals. And all the credit for this checklist goes to the Center for Plant Conservation. This is really their checklist. I just kind of condensed it and reworded it for the sake of this report. So the checklist is written as a series of questions and considerations that need to be addressed at each and every step in the reintroduction process and for a reintroduction project. So using this checklist as a guide should help folks to think more broadly and just consider aspects that they might not have thought of on their own. So I want to close by just reiterating that rare plant reintroductions can be a great tool for conservation. It's important to recognize at the same time that they're very tricky, they're very challenging, and they do often fail, but they are sometimes extremely necessary and successful and very appropriate for a specific species. So our goal here is to just provide this document to be a resource for practitioners and managers and decision makers to have a better understanding of rare plant reintroduction science, so they can have all the information they would need to make decisions on these types of projects. Great, so I want to thank my supervisor, Bob Kartika, and my other colleagues at the New Jersey DEP for all of their assistance and their input in writing this report. And so with that, I'll thank you so much for listening, and I'm happy to take any questions.
Okay, we have a few minutes for questions before Matthew's talk,
yeah.
sorry.
Yeah, I see a question I'd like to address.
Yeah, go ahead and address it. I mean, there's a number of questions in the chat, more than we can get to right now.
Okay, native gardening is becoming increasingly popular. That's generally a good thing. Yes, native gardening, 100% support, Doug Talamay fan. But should there be concern with the use of rare plant species in this plantings? I think yes. Is there a potential to disrupt locally adapted genotypes? Okay, I think my thinking is, my concern with using rare plants in private gardens is, I just wonder, where are people sourcing these propagules? Where are the seeds coming from? Where are the plants coming from? Or are they sourced ethically? So I think that's where my mind goes first. That's my top concern. I mean, if they are sourced ethically, then then the answer is it depends. And yes, I think if you are nearby to native, native population, natural populations of that rare plant, then potentially there could be, how do you put it, disruption of locally adapted genotypes by introducing outpopulation genotypes. Yeah, that was definitely addressed in a lot of the resources that I read. But again, I think, I wonder if the likelihood of that happening, it just depends on where you are. It just depends. Maybe in a lot of the country, the likelihood of that happening is low, but I don't know, it's still a risk. And there's so many wonderful common native plants that can be planted. So, yeah, I guess that's all I say for now for that question. It probably is not a good idea, but maybe in some cases it's less of a concern than other cases. But in my mind, a top concern is, where is that plant material being sourced, and is it being done ethically or not? Okay, here's a question. I know I emphasized the need to have detailed prior information about species habitat and microhabitat needs and their biology and ecology. But yeah, not everything can be known. And a great thing with structuring a reintroduction project as an experiment is that you can test. If there are aspects of the species that are unknown, this is a great way to then test, to learn, what information you're missing. Shouldn't this be a good pathway for species where sufficient propagation? Yeah, absolutely, absolutely. So you learn everything that you can, and there's still unknowns, and then you work those unknowns hopefully into your project design and your experimental design.
Mike, I don't know if you have access to the questions or not, but I have a couple more, Liz, for you.
Okay.
So what are your thoughts on assisted exchange of genes among isolated populations?
It depends. I hate that answer. It does depend, though. I think it depends on knowing what is the genetic structure of the species that you're looking at, and if you can link up with a geneticist. I am not one, certainly, but from my understanding, if you can engage an expert on the topic and figure out, what processes are going to benefit or not that specific species.
Okay.
So yeah, genetic exchange, I mean, it could go in a positive or negative direction. It could lead to inbreeding depression or not. It all depends on, what's going on with that specific species.
Okay. And you emphasized the need for detail for information about the species habitat and microhabitat needs. However, reintroductions can be structured as experiments, allowing us to learn details on such factors as microhabitat requirements. Shouldn't this be a good pathway for species where sufficient propagules are available?
Yeah, absolutely. You can work in your unknowns into the experimental design. And a lot of the case studies that I read did just that, and they learned, so much about the species. And that salt marsh birdsbeak is a great example. They started off thinking that they knew enough, but over the years realized that it was a more complicated species, and it was just through the adaptive management and the continued experimentation and the dedication over the decades that they were able to figure out those unknowns, like the boom and bust of the population and you know, the preferred pollinator, the burrowing bee, and things like that.
Okay. Well, Liz, you know, I'm sure there'll be more discussion, but thank you so much for your presentation today. And I think we'll now turn things over to Matthew Albrecht. Matthew?
Okay, good afternoon. Good early morning to some of you. My name is Matthew Albrecht, conservation scientist at the Missouri Botanical Garden. Let me first thank the State Natural Areas Committee for the invitation to present to the group today. Real pleasure to be here, and that's a great opportunity to share some of our work. So my presentation kind of builds upon Elizabeth's excellent talk, and I'm going to kind of dig into the mechanics of a case study that we've been working on with a federally endangered species, Pyne's ground-plum, Astragalus bibullatus. And in my talk today, I'm going to kind of go over this 2-decade-long reintroduction program and how we're using adaptive management to improve our success rate in this, this recovery program with this endangered species. Before I begin, let me first acknowledge the funders, partners, and collaborators that have been involved in this project. Jeff Call from the U.S. Fish and Wildlife Service has been working with us for over a decade now on this reintroduction program. Many partners at Tennessee Department of Environment and Conservation I'd like to acknowledge, especially David Lincecum, who's really been involved in this project since its beginning. A couple colleagues at Missouri Botanical Garden, Quinn Long and Noah Dell, who've been working on implementing this project with me. And finally, I'd like to acknowledge Dr. Kim McHugh, who's now the Chief Science Officer at Desert Botanic Garden, who really initiated this reintroduction program back in 2001 when she was a conservation scientist at the Missouri Botanical Garden. There are lots of people who've been involved in helping this project, field assistants, volunteers, some of whom are on this call. I want to thank you for all the time that you put into this project. Too many people to list on a single slide. So let's set the stage for Pyne's ground-plum. And this is again, a federally endangered species. That's one of a number of at-risk plant species that's endemic or near endemic to Nashville Basin limestone glade and woodland ecosystems. And this ecosystem is really a grassland ecosystem that consists of a mosaic of plant communities, ranging from kind of sparsely vegetative rock outcrops to deeper soil barrens, to savannas and woodlands. And it hosts a number of rare plant species that I'm showing you at the bottom of this slide, including Pyne's ground-plum. And obviously by its name, Nashville Basin, this particular ecosystem is pretty much isolated in central Tennessee, mostly in and around Nashville metropolitan area. So Pyne's ground-plum was listed as federally endangered back in 1991. And it was listed as endangered due to the small number of known populations in the landscape. And a majority of the global population of the species is distributed along about a 1.5 mile stretch of county roads south of Murfreesboro, Tennessee, and Rutherford County. And so if you look at the bottom right-hand corner of the map, there, that shaded box shows you the extant occurrences of Pyne's ground-plum. So this is a species that's endemic to a single watershed, the Stones River Basin or the Stones River watershed. And from historical specimens, we know that this species was potentially once more broadly distributed throughout this watershed. And so you can see some points where we know of historic localities where the species once occurred, including a site that's now inundated by the Percy Priest Reservoir, which was a dam built along the Stones River back in the 1950s. So most of the known populations that exist today, many of them are quite small, known from less than 100 individuals. Several of them are unprotected private property. And so really only about half of these can be considered protected viable occurrences in the landscape. And, you know, given this is such a highly fragmented and urbanized region, it's highly unlikely that many new populations will be discovered of Pyne's ground-plum. This is also a very well botanized region for the past century or so due to its unique flora. And so reintroduction is being used in the recovery plan to help meet the recovery goals for downlisting and delisting for the species. And so to reclassify this species as threatened will require 11 viable protected occurrences, and to ultimately delist this species will require 16 viable occurrences. And so the goal of this program is to expand the distribution and abundance in the historic range of the species in order to reduce the species extinction risk and improve its conservation recovery. So as I mentioned earlier, this program has been ongoing for almost 2 decades. I've been involved in it for a little over a decade now. Let me just kind of take a step back before we dive into some of the details and give you sort of a broader brush overview of what this program looks like. So at the Missouri Botanical Garden, we've been collecting seeds of globally at-risk plant species throughout the lower Midwest and the Mid-South for the past almost 40 years. We've been building up seed collections of at-risk species like Pyne's ground-plum. Our first collections for Pyne's ground-plum in our seed bank go back to the early 1990s. And so over the past 3 decades, we've been slowly and sustainably collecting small quantities of seed from natural populations and building up genetic diversity in the seed bank. And we're using that seed in our frozen seed bank as the source material for this reintroduction program. We're following the CPC best practice guidelines. When we collect seed, we're collecting by what we call maternal family. So we keep all the seeds from each individual plant in the wild separate. And when we remove them from the freezer, we track them. Each seed is given a unique identifier, and we track it from the freezer to the field. So it's a very extensive sort of demographic study. Over the past 2 decades, we've outplanted over 1,200 greenhouse-grown plants. We've worked on developing and refining propagation protocols over the past 2 decades for the species. Like many rare conservative species, this is a challenging species to grow in propagation. It's taken a lot of experimentation and refinement to successfully grow this species. We've outplanted those 1,200 grown plants across 7 designated natural areas that are managed by the Tennessee Department of Environment and Conservation within the historic range of the species in the Stones River watershed. And you might be thinking, well, 1,200 plants doesn't sound like that many, but if you think about the global population of the species as being around probably about 5,000 individuals, you know, we're increasing the population size, global population of this size by about 20%. So, and given the challenges of such a rare species, given that small populations produce so little seed materials in such short supply, it's difficult to propagate a large number of plants at any given time. And I'll talk a little bit more about that propagation process as I move along this this talk today. So some of the sort of general principles that we've been following in this reintroduction program follow those that are outlined in the Center for Plant Conservation's best practice reintroduction guidelines. So in this case, we're using transplants rather than seeds as source material for our reintroduction program. And that's simply because seeds, like for many at-risk species, are incredibly rare, incredibly valuable, and incredibly precious. And so throwing out seed as the source material at our reintroduction sites would be extremely costly for us in the sense that seeds are just so uncommon. And let me give you an example. We've been demographically monitoring 5 natural populations of the species for over a decade now. And if we look at seedlings that have come into these populations, we've tagged them and we follow their demographic fate. Of about the 550 seedlings that we've tagged, only about a half a dozen of those have ever gone on to become flowering, mature individuals. And so there's an incredible bottleneck there between a seed going from a seedling to a mature flowering plant. And so if you imagine starting your reintroductions from seed, you'd have very low success rates. So we sort of overcome and bypass that bottleneck by propagating plants at the Missouri Botanical Garden and then bringing out those seedling transplants to our reintroduction sites. We're also trying to create large genetically diverse founder populations. We know that greater genetic variation in our populations means more resilience to perturbations such as drought, other extreme climatic events associated with climate change, other sort of environmental disturbances that might happen in the population. We're also increasing the chance of having compatible mates in the population. This is probably a species that is at the very least a facultative outcrosser, so it needs compatible mates in the population to successfully cross-pollinate. And so by having large genetically diverse founder populations, we're increasing the chance of reproductive success. Similarly, we're also focusing on trying to have founder sizes greater than 50 individuals. And I'll explain a little bit more about that in a moment, because some of our early reintroductions might have been failing because we had small founder sizes. And following the CPC guidelines in all of our reintroductions, when we pull our seed accessions out of the freezer to propagate them, we do our very best to try to evenly distribute those maternal lines. So we have more or less genetically even representation of individuals at our reintroduction sites. And this is designed to help reduce eventual genetic bottlenecks that may happen with limited genetic diversity. So I think probably one of the most challenging aspects of a rare plant reintroduction program is dealing with what source populations to use and dealing with the genetics. And just by looking at the chat from Liz's talk, I think that's probably true. A lot of the questions sort of dealt with how far should we be removing plant material in our reintroductions? What are some rules of thumb for that? And really, it's a case-by-case basis. And let me show you what we've been doing with Pyne's ground-plum. So I think when we're doing restorations, we have to first confront the question, do we mix our source material together, or do we use single population sources when we're designing reintroductions? And again, that's going to depend on the study system, the species, the project goals, and what you might know about the genetic variation and structure in that species. Fortunately for Pyne's ground-plum, we've had several genetic studies that we can draw from to understand how genetic variation is partitioned within the species. What I'm showing you on the right-hand side of this graph is some work from my collaborator, Ashley Morris, who looked at microsatellite data in Astragalus, bibullatus across natural and reintroduced populations. And what she found was consistent with some earlier work that was done by Carol Baskoff looking at AFLPs and isozyme markers, in that this species has most of its genetic variation distributed within populations and not among populations. So those FST values I'm showing you at the top of that graph, that 0.007 to 0.056, that's telling you what the genetic differentiation is among populations. And so it's pretty low for this species. So that tells us that historically, there was likely gene flow among populations, and that most of the genetic variation is distributed within populations. And we've been using this to design our reintroductions around mixing population sources together to try to maximize the amount of genetic diversity we have in our founding populations. You'll notice there's one population in this diagram. There's a circle in the left-hand corner of this map that shows N-RV. That's rockvale. That's one of the more recently discovered populations of this species that's sort of isolated from the others, and it does appear to have a little bit more genetic differentiation from the core populations that are in very close geographic proximity to each other. We could talk all day about propagation, but let me give you a few of the sort of key points and take-homes from our work with Pyne's ground-plum. It's really important during cultivation that you do not favor the most vigorous plants. You want to have diversity, and so we're not growing plants to take to Home Depot to have the best looking plants to sell. We want variation, both phenotypic variation, we want genetic variation. And just because the plants may not look that robust in cultivation doesn't mean they're not going to perform well in the field. And so we're very careful to not select for vigorous plants during cultivation. We also have a protocol for screening plants for pathogens and pests before we bring them out to the wild. We have expert horticulturalists who are trained to examine plants for plant disease and pest, and we treat any plants that are infected. And in the past, we've actually withheld plants that have been infected from bringing them out in the wild. So we're ensuring we're not cross-contaminating anything in the natural populations. So let me just begin to dissect this reintroduction program, again, that began back in 2001 by my predecessor, Dr. Kim McHugh. And the first round of reintroductions occurred at a location called Stones River National Battlefield. This is a site that's managed by the National Park Service. It's in the city limits of Murfreesboro, Tennessee. And it was selected for a couple of reasons. One, it was protected habitat. Two, it was in relative close proximity to most of the naturally known populations at that time. And three, there was what was presumably suitable glade habitat. And what I'm showing you in this figure is aerial view of the battlefield. This is the site of a famous historic Civil War battle. You can see these openings in the forested landscape here. These are these rocky outcrop openings, and the sites located, labeled with arrows, are locations of reintroduction sites that were chosen at the beginning of the study. And those sites were chosen because, by and large, they thought that the edges of the glades offered suitable habitat and were somewhat similar in appearance to the few known populations the few populations that were known at the time that this project started. And this project was designed as an experiment. So we knew very little about Astragalus bibullatus at the time of this reintroduction. We knew very little really about the biology of the species and restoration techniques with the species. And so this project was designed to really answer two questions. One, of the natural population seed sources we're using, are there some populations that seem to maybe perform better at novel reintroduction sites than others? And two, what is the best time of the year we should be transplanting greenhouse grown seedlings? Should it be fall or spring? So here I'm showing you graphs that show the survival of the 111 individuals that were outplanted across these 5 glade sites back in 2001. And this is showing their survival rate over a 7-year period. In that first graph, you can see there's really no difference in survival rate of seedlings that were derived from the 3 different natural population sources. And this sort of matches the genetic data that there didn't really seem to be any genetic differentiation among those populations. And it didn't really matter where you came from, you had an equal chance of surviving. So there's no effective population seed source. But what Kim did find is that those individuals that were planted in the fall had much higher survival rates than those planted in spring. And so through this experimentation, they were able to elucidate some key factors that might guide future reintroductions in the species. When I came on board this project in about 2009, 4 of the 5 sites had essentially gone extinct. The founder individuals had basically not survived over this time period, and there was not enough second generation recruitment to replace those individuals, with the exception of 1 site where there were a handful of new second generation recruits derived from those founder individuals. And as we started to pick up the pieces of this reintroduction program and move it forward, we had to kind of wrap our heads around why these reintroduced populations went extinct. And when we went out to look at natural populations to develop a demographic study, one thing that struck me was how different the natural populations that were known at the time looked and their habitat condition. And so here I'm showing you the 3 different types of habitat conditions you could find Pyne's ground plum in. You can find them in really open conditions. So the site on the left-hand side, this is at Flat Rock, Cedar Gladed Barrens designated natural area. This is what we call the airport site. This is the site essentially under a power line cut, and it's been kept open through power line right-of-way maintenance and through periodic bush hogging by the Tennessee Department of Environment and Conservation. This site harbors the largest population of Pyne's ground plum. The sites that seem to be most typical of this species were sort of these deeper soil pockets around the edges of these really shallow, thin-soiled rock outcrop glades. And the flags I'm showing you in that middle picture are locations of Pyne's ground plum. And again, I probably should have mentioned this before. This is a very small statured rosette-forming perennial, so it's not a tall species. And so you'll often find it growing in bands, these sort of microbarrens that have C4 perennial grasses growing around the edges of these glades before they abruptly transition to hardwood forest. And finally, there are a couple unusual sites that are basically openings in an otherwise forested kind of landscape. And these are thin-soiled rocky outcrop openings. And here again, you can see flags next to plants that we're monitoring. And so then the question becomes, what are the habitat conditions that support viable populations of the species? And we really didn't have an answer to that question at the time. So through our demographic surveys, we began looking at how microhabitat variation related to the demographic structure of the species. And what we found is that basically the growth and reproduction of the species was influenced by the amount of woody vegetation growing above it. And so here you can see tree canopy cover on the x-axis, and we see plants growing, producing more stems and having greater growth rates at lower canopy cover. In the middle graph, we find more plants flowering at lower canopy cover, and we find more fruit production in plants growing in more open conditions. And so this got us thinking that maybe the habitat's just not quite right in those initial reintroduction sites, and maybe they're too shady and edgy to support growth and reproduction, which is what you need in a reintroduced population at the initial stages to sort of jumpstart that population growth. You want plants growing, transitioning into flowering individuals, and producing seed so you can jumpstart that next generation and facilitate population growth. So we put our heads together and we said, well, maybe the habitat conditions weren't right in that last reintroduction, but there are a number of other things that could have also been sort of influencing those reintroduction outcomes. And we began to really formulate our program around an adaptive management cycle. And that's what I'm showing you here in this slide. And so if you're sure most of you are familiar with adaptive management, but it's a really great way to sort of frame a reintroduction program, because adaptive management is designed to reduce uncertainty in your study system. And if you think about rare plant species like Pyne's ground plum, most of the time we're going to start a program where the species biology is poorly understood, or its response to management is uncertain or unknown. And this is in part because we're just dealing with such incredibly rare species. These are difficult things to study. We also maybe don't have time to do the experimentation. Maybe we don't have the propagules to do the sort of proper control, replicate experimentation to answer some of these questions. Adaptive management allows us to answer these questions as we move through our recovery and reintroduction program. So in this case, let me just quickly walk you through some of our thinking here. So our problem is, well, what is suitable habitat for this species, and how can we reintroduce and create populations that are going to persist over time? We've developed some predictions. We think more open habitat with less woody encroachment is favorable for this species, but there may be other things that might be operating. For example, in the first round of reintroductions, the founder population sizes were quite small. There were only about 30 or 40 individuals. And in any given year, only about 20 to 25% of those plants will flower. So when you have a small population size and you have few plants flowering in any given year, you're reducing the chances of successful cross-pollination, and you're reducing the chances that pollinators are going to be moving pollen from one genotype to another, and you're going to reduce the chances of having successful seed production. So if we increase the population sizes, our prediction is that we may have greater growth, flowering, and reproduction in these reintroductions, and that might in turn improve long-term success. And so we can design a study, a reintroduction study, to test these ideas, and then we can implement them, we can monitor their outcomes, and then we can reconvene and update our thinking about this study system. And then we can redefine new problems that might emerge, and we can continue to go through the cycle to improve success. So what we did in our next round of reintroductions, what I'm calling experimental reintroductions 2.0 for lack of a more creative term, is we went through and we changed some of the variables in our design. So we knew from the first round of reintroductions that we needed to transplant during the fall, and we're going to get greater success. We increased the genetic diversity of our founder populations by using more source population material. We also increased the number of individuals in the founder population size as well. And again, with the hope of overcoming some of those barriers to small population size. We also decided to increase the extent of microhabitats in which we were planting individuals to try to increase the chances of matching the correct genotype to the correct microhabitat. And then we did management at these sites. So we opened these sites up to reduce woody encroachment, which we know is a potential threat to the species. And then we also manipulated access to herbivores by caging half the plants. And there were some anecdotal observations in the first round of reintroduction that deer, which are quite abundant at Stones River National Battlefield, may have been browsing plants early on, and that may have in turn influenced their long-term survival and growth. And so we went back to Stones River National Battlefield, to a site where we had some success, where we saw second generation recruitment. We did management at the site. You can see with the blue arrow, one of the many cedars that we cut out of this area to sort of increase light availability. We chose another site that's a designated natural area that has reintroduced populations of Echinacea tennesseeensis, Tennessee coneflower, which Elizabeth mentioned in her talk. And then we started thinking out of the box and we said, you know, I think maybe the historic habitat of this species is a little bit different than what we see in natural populations, and that over time, natural populations have become much more woody encroached and much more degraded than probably what they were historically. And so we chose a site called Couchville Cedar Glen and Barrens, which is a site that's been managed periodically with fire in the past and has a more open grassland-like landscape around the rock outcrop openings. And so we chose this site as a site to sort of test this hypothesis that maybe this more xeric barren habitat maybe more matches the historical habitat of the species. And so interestingly, what we found is that in our Stones River National Battlefield reintroductions, despite increasing habitat openness, despite reducing herbivores through caging, despite increasing the founder size to overcome small population size issues, we still found low population viability at this site. We found that plants there had survival rates that were 2 or 3 times lower than those at our Couchville site, which again is this more xeric barren site. And if you look at flowering and fruit production, we find way more fruits being produced at Couchville than we did at Stones River National Battlefield, even for those plants that were caged. So something else was going on at Stones River National Battlefield. The habitat's not suitable for this species at that location. And so we began to start to look at some other factors in this study system. And so the thinking was that all of the sites we've been reintroducing the species to occur on what they call Gladeville soils. But it turns out Gladeville soils can occur on a couple different geologies in this Nashville Basin. And there are 2 sort of primary limestones, Ridley limestone and Lebanon limestone. And Ridley limestone is shown here in the purple. And all of the failed reintroductions with Pyne's ground-plum turn out have all occurred historically on sites that were underlain by this Ridley limestone. And this type of limestone is a more interbedded limestone, and it's less fragmented than the Lebanon limestone. And so sites with Ridley limestone tend to have a slightly different hydrology. They tend to hold water more, and they're less well drained. Whereas the Lebanon limestone has this highly fractured bedrock that creates extremely drought-prone soils. And you can notice this when you walk out to a glade after a precipitation event, and you walk through a glade that's underlain by Lebanon limestone versus Ridley, and you'll notice a difference in the sponginess of the soil. And so if you look at natural populations, most of them are sort of distributed on Lebanon limestone. Those are those small red dots in this picture. But if you are at the edges of that interface between Lebanon and Ridley, and so this guy's thinking that really this is a species that probably can only occur on this Ridley limestone and I'm sorry, Lebanon limestone. And when you look at the distribution of Lebanon limestone in the Nashville Basin, it forms this ring around Ridley limestone, and it's really less common than Ridley limestone. And if you start looking at the distribution of many of the at-risk species that are in decline, most of them are largely associated with this Lebanon limestone, this highly fractured limestone that also creates highly successional sites. The fractured bedrock creates lots of deeper soil pockets that Pyne's ground-plum and other at-risk species might depend upon, but they're quickly colonized by woody vegetation in the absence of disturbance. And so it turns out through the second round of reintroductions, when we go back and revisit our adaptive management cycle, that we've learned a couple things about the species that are going to hopefully improve our success rates in the future. The first is that this species has an extremely narrow habitat requirements. It can only probably exist on this one particular type of Lebanon limestone. It can only exist in deeper soil pockets on this Lebanon limestone. And another thing that we discovered in this reintroduction program that wasn't known prior to this in our monitoring of natural populations is that herbivory turns out to be a pretty pervasive threat to this species. And so here I'm showing you the number of fruits produced on reintroduced plants across these different limestone types, plants that have been caged and plants that have been uncaged. And basically plants that are not caged in our reintroduction program hardly flower and rarely ever produce fruits. And so I'm showing you in these pictures on the right-hand side of this graph, of the slide. I'm showing you plants in the upper image that are not caged. And you can see the plants are about a quarter of the size of those plants that are literally growing a couple feet from the plants in the cage. And you can see the plants in the cage are much larger. They're flowering, and those plants will go on to produce fruit. And so repeated herbivory events, largely through deer and rabbits, basically knock this perennial back and prevent it from transitioning to become flowering individuals. And so herbivory is a serious threat, and we're finding this across a lot of the different natural areas that we're doing our reintroductions with. So we've learned a lot from this particular adaptive management cycle. We've learned things about the transplant season. We've learned things about source population performance, the species microhabitat requirements, its broader habitat requirements in the landscape. And now we know that herbivory is a serious threat that really wasn't known before prior to this program. But we also got to thinking too, as we expanded our reintroductions across the landscape, we also noted quite a bit of variation in plant survival and plant performance. And this got us thinking about what other factors might be influencing reintroduction success. And so I had a graduate student, Rachel Beckman, who I co-advised along with Dr. Scott Mangan at Washington University on a project to look at how the soil microbiome may be influencing the species. So if you look at a lot of slow-growing conservative species in grasslands, many of them tend to have obligate relationships with arbuscular mycorrhizal fungi. And they have dependencies more so than early successional species on soil microbiota. And so to sort of test this idea, what Rachel did is she grew Pyne's ground-plum on soils that she collected from natural population sites labeled here as historically present glades. And then she collected soils from sites that we had planned to do our reintroductions at in the future. And she conditioned those soils by growing Pyne's ground-plum in them. And then she took those conditioned soils and she sterilized half of them, and then she regrew Pyne's ground-plum in them to see how Pyne's ground-plum would perform. And if you look at the historically present glades, you can see when you sterilize the soil and you remove microbes, you have a reduction in the growth rate of this species. And so that suggests there's some dependency on soil biota that this species has, and without that soil biota, it has reduced growth. Interestingly, when you look at the historically absent sites, again, these are sites that the species is not known to occur in modern times, but these are sites that appear to be suitable, have suitable habitat for the species and are sites slated for future reintroduction. Overall, the plants performed lower than they did. They had less growth rates than plants growing on historically present glade soil, and you don't get this signal between live and sterile soil, suggesting the microbiome is different. It's not responding to the microbiome at those sites. She explored this a little bit further, and she did what's called a plant soil feedback experiment. I can't go into the details due to lack of time, but I'll point you to a paper she published in the Journal of Restoration Ecology last year. And if you'd like a copy of this, please feel free to contact me offline. I'll be happy to give you a copy. But what she did is she took two common species that Pyne's ground-plum grows with. One is Tennessee milkvetch, Astragalus tennesseeensis. This is a congener that's a little bit more abundant than Astragalus bibullatus, and little bluestem, which is a common C4 grass in barrens and grasslands within the Nashville Basin. And again, she did this feedback experiment where she conditioned the soils with these different species, and then she grew Pyne's ground-plum on soils that had been conditioned by these two other common species. And what she found is that when she grew Pyne's ground-plum on soils conditioned by little bluestem, those plants tended to produce less nodules, rhizobia nodules, than plants grown on soils conditioned by itself or by the closely related Astragalus tennesseeensis. She also found overall greater nodulation on soils from natural populations than soils collected from historically absent sites. And so this is sort of illuminating because it suggests that Tennessee milkvetch could serve as an indicator species for habitat that might be suitable and the soil microbiome might be present that Astragalus bibullatus can associate with, and that in turn might improve the success rates of our future reintroductions. And so one of the things we're doing now in our reintroductions is we're basically inoculating our plants with small amounts of rhizobia and field soil to give them the microbiome they need to associate during the propagation stage. And then we're looking at sites across the landscape that have Astragalus tennesciensis present. And we're looking at sites that are only found on Lebanon limestone. And we're focusing on sites that have a history of management for woody encroachment and have prescribed fire management plans to keep woody encroachment in check at these sites. So we've, with our partners at U.S. Fish and Wildlife Service and TDEC, have really extensively surveyed sites and located about 7 different designated natural areas that sort of meet these criteria. And over the past 5 years, we've been reintroducing Pyne's ground-plum to these designated natural areas with this sort of newfound information that we have on the biology of the species. And in the short term, our results are much improved over some of our previous reintroductions. We're finding 80 to 95% survival of founder individuals over a 3-year period, whereas before we were finding much less, typically only about 50 or 60% survival, even in the sites that we consider to be our best sites. We're finding overall greater fruit production and reintroduced populations relative to the natural populations. And we've also chosen sites that are connected to previous reintroductions and to natural populations to try to improve connectivity. So we're focusing on sites that might help facilitate gene flow between populations of Pyne's ground-plum, because the genetic data suggests historically gene flow was important in this system. So something Elizabeth mentioned in her talk, and something that's certainly a challenge with Pyne's ground-plum, is really having enough material to conduct your reintroductions. And so what we've developed with Pyne's ground-plum is a seed amplification program to increase genetic diversity and the amount of material we have available. And so we're doing this at our Shaw Nature Reserve, where we're basically taking material out of our seed bank, we're propagating it, and then we're allowing it to open pollinate in a common garden setting. And then we're taking that seed that we're collecting from this common garden, we're using it in our reintroduction program. And so that's how we're sort of bulking up material to conduct this experimental program. So finally, let me kind of take a step back and talk about some broader lessons learned that might be applicable to other reintroduction programs. I think that successful reintroductions may require going through this adaptive management cycle several times for species. And I don't think many reintroductions will be successful on just single attempts. It's often going to take experimentation and learning as we go, and adjusting our techniques as we learn about the biology of the system. And so I think by going through these AM cycles, we're going to improve our outcomes in the future. Something else Elizabeth mentioned that I'd like to also sort of illuminate, and that's that partnerships and sustained funding are really critical. We've been fortunate in this program to have wonderful sustained partnerships with the Tennessee Department of Environment and Conservation, U.S. Fish and Wildlife Service, both for funding support, logistical support, assistance in the field, and really partnering together to talk about habitat management for this species and other at-risk species in this system as well. And finally, something that I've come to discover with this program is that I think species conservation can actually inform ecosystem management and conservation. So prior to this reintroduction program, most of the natural populations of Pyne's ground-plum were not being managed with fire for woody encroachment, but through demographic surveys, through additional research, through reintroduction experiments, as we've learned about this particular species, we begin to understand that historically, these deeper soil pockets within these glade mosaics were probably kept open by disturbance processes such as fire and other factors. And so I think since this program started, fire and woody vegetation encroachment management has become widespread across multiple natural areas in this system. So it's not just benefiting Pyne's ground-plum, but it's benefiting a number of at-risk grassland species that I think depend on these ecological processes. that's another important take-home point. And with that, I'm about out of time, so I will wrap it up. Thank you for listening.
All right, Matthew, we've got a few questions here in a few minutes to dive into these. Let me, one of the questions was research looking into the primary pollinators of these plant species and the relationship that might be there.
Okay. Yeah, so that research is ongoing by a group of students and professors at Middle Tennessee State University. We've done some preliminary work. So this is a early spring flowering legume. The pollinators tend to be generalist pollinators in the system. So again, they've been looking at the breeding system, and they just started this study to really look at pollinators in more detail. We've actually set up camera traps at a couple different sites that we've been looking at identifying pollinators as well. So that's sort of research that's in motion right now, and we should have results over the next couple of years.
Okay, and then another question regards, have you considered planting seeds directly when they are ripe on site?
No, because of the extremely low germination rates and seeds, and simply the species has physical dormancy and forms a persistent soil seed bank. So if we planted seeds that we hadn't broken dormancy in, it may take years for them to actually germinate. And until we get a handle on the sort of seedling recruitment bottleneck in this species, we'd have pretty low success rates in our reintroductions so far. So that's why we've been focusing on seedlings.
Okay, and then the cage design that you used, was that mainly to prevent deer herbivory or rabbit herbivory and rodent herbivory? And then how tall were those cages?
So those cages were designed to prevent all of the above.
Okay.
Simply because we actually didn't know what was all the different browsers in this system. And we think there are multiple browsers. And in fact, again, we're using camera traps to better understand the identity of herbivores and the abundance of those herbivores. We certainly have caught deer on camera eating plants, but based on the browse patterns, we also think rabbits and potentially maybe some Peromyscus might be involved in snipping stems as they emerge early in spring. So the cage design is hardware cloth. It's the quarter diameter openings that basically prevents rodents from accessing the cages. They're about 3 feet tall, and I can give you exact dimensions and how we constructed them if they want to contact me offline.
Okay, sure. And then the last question here, camera trap protocols for pollinators. Are there any standards out there?
It really depends on what you're after and your study system. So I can share the design of our camera trap protocols for pollinators. So it requires cameras that can take up close, you have to really have cameras close to your focal plants. We've used lenses to magnify the images. I can talk more offline about that because there's a lot that kind of went into that. and
Sure.
there are some papers out there that have experimented with this.
Okay. Well, I think at this point, we will load up our stewardship shorts, and then after that, we'll have a more open discussion and wrap our program up.
Hi, I'm Margie Hunter, a volunteer with the Trifolium calcaricum Safeguarding Project in Tennessee. In the late 1800s, Dr. Augustine Gattinger documented this clover as common in the rocky limestone glades of Middle Tennessee, but he misidentified it as Trifolium stoloniferum, which is not found in the state. The Tennessee and Virginia populations were discovered a century later and declared a new species in 1992. Running glade clover produces flowers only at the end of the stolon. It does not have leaflet markings, and its flowering stem is leafless. These characteristics differentiate it from the similar non-native white clover and the native running buffalo clover. Characteristics specific to running glade clover include maroon veins on the floret standard, long attenuated calyx lobes, and leaf-like stipules. With a G1 rank, Worldwide distribution consists of Lee County, Virginia, and Rutherford, Bedford, and Marshall counties in Middle Tennessee. Plants collected from the Rutherford population were outplanted in Wilson County to the north. At the top right, 18 separate outplanting sites are located within 3 primary areas in Cedars of Lebanon State Park in Wilson County. Just below is the natural population in Rutherford County, the Bass property, the source site for the plant material. A small parcel adjacent to the Bass property and containing the clover is now owned by the state, but the vast majority of the plants remain on private land. At the bottom left, Wilson School Road State Natural Area in Marshall County and the privately owned Parsons property in Bedford County contain natural populations. State-owned property at Henry Horton State Park and Howard Bridge Glade are potential outplanting sites for collections from the Parsons tract. Safeguarding efforts took place on a shoestring budget behind the maintenance building at Cedars of Lebanon State Park. We conducted 3 collections and 3 outplantings between July 2016 and December 2018. Evidence of success followed quickly as stolons slipped through the cages. Most plant groupings remain caged, but a small number are not. 2 surviving plants in this grouping show excellent spread. They produced 5 flower heads in 2020 and 8 in 2021. The species' clonal spread has made it impossible to continue tracking individuals, which were planted mostly in groups of 3. Of the 93 groupings, 73 are currently rated good or robust, primarily on subjective evaluation of their appearance, the crown foliage, and overall spread. Plants given to Cheekwood for safeguarding were collateral victims of COVID when the garden shut down in 2020. We've learned a few lessons. Starting with larger plants improves success. We've outplanted in both late fall and early spring. The spring plantings seem to perform better. Overwintering in a cold greenhouse allows more time for growth and eliminates the weak plants. Winter sun is likely a factor. The overall habitat is ecotonal, light to moderate shade in woodlands of the limestone cedar glade complex. In the right-hand photo, the plant is growing in the shady margin. Good ambient light is important, particularly for flowering, and we are looking closely at the value of that winter sunlight since it is evergreen. We established 52 plots at the 3 natural populations and collected a variety of environmental data and floristic community data, which we will analyze this year. We hope this study will inform management strategies we can implement and evaluate in the future. There is much we still do not know and lots of opportunities for additional research. I wish to thank the Tennessee Plant Conservation Alliance and the Tennessee Department of Environment and Conservation's Division of Natural Areas for their support of this project, and the Natural Areas Association for the opportunity to share it. Thank you.
Good afternoon, everyone. My name is Doug Manning. I'm the biologist at New River Gorge National Park and Preserve, also Gauley River National Recreation Area, and Bluestone National Scenic River. Today I'm going to be presenting a short talk on safeguarding, propagation, and outplanting of Virginia Spiraea on NPS land. Quick background on Virginia Spiraea. It's a disturbance-adapted clonal shrub that grows in the central and southern Appalachians. It tends to grow in these scour zones, these areas along rivers and streams that receive periodic flooding to the degree that it actually removes a lot of woody vegetation, such as trees and other species not adapted to handle those growing conditions. In 1990, it was listed as threatened under the Endangered Species Act, and those threats include habitat loss from things like development, invasive species encroachment, and hydrological modifications such as dams. It also suffers from some potential reproductive issues linked to polyploidy, which are being investigated right now. The National Park Service is proposing to do Virginia spirea recovery work at 2 of our 3 parks. The first of which is Gauley River National Recreation Area. This is actually the world hotspot for Virginia spirea. The park contains portions of the Gauley and Meadow rivers. And these rivers are relatively high gradient and promote the conditions for these scour communities to develop. This periodic flooding promotes not only habitat for Virginia spirea, but actually good habitat for a lot of other rare plants that are dependent on these conditions. We have lots of state tract species as well as some G2 plant communities that occur in the river or along the river. However, one of the issues we anticipate is a lot of really difficult access. Bluestone National Scenic River is the other park that we're planning to do Virginia spirea recovery work in. There's actually there are no extant occurrences of Virginia spirea within the park boundary. However, it does occur just upstream. This park as well has some of these rare plant communities and rare species that are in these scour maintained sections of the river. And also, again, the nature of this park is that it is relatively difficult access. So what is the National Park Service planning to do, actually? We are going to be assisting the West Virginia Department of Natural Resources in monitoring extant occurrences throughout the parks. We'll be conducting habitat manipulation, principally invasive plant removal, as demonstrated here on the right with one of our interns from last summer who was actually treating invasive plants along the Bluestone River. And we will be also reducing or improving light availability in certain cases where Virginia spirea is limited by trees and shrubs overtopping it. We will also be propagating Virginia spirea from cuttings at Mount Cuba Center. These cuttings will be used to augment declining occurrences in the Gauley River National Recreation Area, and as well as an experimental outplanting in Bluestone National Scenic River. An important note, genetic fidelity is going to be maintained to the watershed for Bluestone, and in Gauley, we will be genetic fidelity will be maintained to each augmented occurrence. We will, of course, be monitoring and reporting on these augmented and experimental plantings. And Mount Cuba Center will be holding some specimens in permanent cultivation as well at their botanical garden. All of these proposed actions, I wanted to note, fall within the scope of the Fish and Wildlife Recovery Plan for Virginia spirea. And that kind of wraps it up. Really brief summary. Really had to obviously gloss over a lot of things, but I have time now to take any questions that anyone might have. And yeah, thank you for your time.
Okay, great. We still have some time here for some final questions. Just trying to go back through here. I think we've covered most of these. If anybody wants to chime in with any more questions, please do so at this point.
Mike, just a reminder too to everybody that both presenters of the stewardship shorts are in the audience, so they're happy to answer questions about those 2 short videos.
There is a question here. The CPC guidelines for when to do reintroductions seem to be really restrictive. However, given how long it can take to figure out how to do a reintroduction successfully, as shown with Pyne's ground-plum, should we be starting this process earlier on in the species decline, especially if we are introducing to a site without existing populations? Seems like the risks are low and the rewards are high. And a follow-up is primary objective is to protect the wild populations and figure out why they are declining first and not to lose those, which makes sense. Matthew and Elizabeth, do you want to chime in on those?
Yeah, I'll chime in. I think that's a great point. I think the CPC guidelines are restrictive for the reason that reintroduction is not used in mitigation for mitigation purposes. And You know, reintroductions, when they're designed as experiments, like what I outlined with Pyne's ground-plum, can go a long way to answering questions about the biology of the system and of that species. So I think they're restrictive as well, because we want people to think carefully about the broader conservation and recovery strategy for that species, and to not rush into doing a reintroduction when we haven't thought about protecting natural populations, we haven't thought about managing natural populations, we haven't really thought about a lot of these other factors. So it may come across as being restrictive. I think if you dig into the guidelines a bit more, you'll see how reintroductions can be used to sort of reduce this uncertainty and unknowns that are bound to pop up with many of these at-risk species that we're all working with.
Thanks. In one of the questions, maybe I saw it earlier, a reference to climate change and assisted migration. And I think that opens up, well, that'd be a whole other discussion. A whole other set of questions, especially with rare species. Oh, here's a question regarding the ground plum. Are there any soil or rhizobia inoculation treatments for outplanting? And I think you might have discussed that if I'm remembering right. But anyways, have you guys been tracking that with survivorship, et cetera?
We have not tracked that experimentally in the field. We've done some greenhouse studies where we've tracked that work, and we're sort of basing this on some of Rachel's work, where she showed this sort of dependency on soil microbiota in this species. So no, we haven't published anything specifically looking at reintroduction experiments with and without inoculation. But that would be something we could certainly do in the future.
And I guess to answer the climate change question, I guess seed banking is probably our best bet at this point, where we at least preserving propagules, given our limited knowledge of of how things are going to change and you know, the different climate envelopes. I do believe that. So somebody at the botanical garden is modeling this, isn't it? John Smith, perhaps.
Yeah,
that was some of his research.
Adam Smith has done some species distribution modeling with a number of risk species in the region. You know, there are many challenges with SDMs with rare species, both in terms of getting enough data points to actually build a model, and the fact that many of the at-risk species that we've been working with are really edaphic specialists and edaphic endemics. And so there are another suite of features that are restricting their distribution in space rather than just climate. So we've been using our long-term demography data to ask questions about how climate extremes might be influencing long-term persistence and population viability in the species.
We have some Spiraea questions. Are known Virginia Spiraea populations reproducing by fruit or only by vegetative reproduction, or both, or neither? And a similar question on the Spiraea in Tennessee.
Yeah, sorry, my video is apparently not working today.
Oh, that's okay, Doug. Let's just, we can hear you just fine now.
Okay, great. So we are choosing to go with cuttings as our vegetative propagation source material because Virginia Spiraea has, it's often has very low seed set, and a lot of studies have demonstrated that often it has extremely low seed viability. And the other factor to consider for us with regard to that is I mentioned a little bit in the briefly in the presentation that there's some issues with polyploidy. There have been some studies coming out right now that are addressing this, and there are concerns over some sterile individuals out there, and we certainly have some issues with like triploid Spiraea within our parks. So in order to ensure that we wouldn't have any issues with mixing genotypes, we are strictly going to stick to augmenting using propagules taken from the source cuttings. And I'm happy to share some of the publications, if anyone who's interested, that have kind of studied those issues surrounding the outcrossing and genetic viability surrounding Virginia Spiraea.
Okay, yeah, that'd be fantastic. Maybe we could post your email on here and then folks could contact you. Well, I think we're just swimming in information here. And so lots to absorb. Folks, if you want to unmute and have any more discussion, I personally am out of ideas, just a lot of information to absorb at this point. But would sure appreciate any feedback on, like we said, on future roundtables in particular. Please let us know of different topics that you might want to see addressed. Lisa, any closing thoughts or remarks?
Mike, I don't think so. I just want to take this opportunity to thank all of our presenters. This is a topic that clearly is of interest to so many practitioners working on ecologically significant landscapes and with plant species of concern. So I just, again, want to thank everybody, Liz, Matthew, Doug, Margie. We really appreciate your contributions today, and we'll keep this topic front of mind and love hearing back from all of you. Again, Natural Areas Association exists to serve you and your work. So please keep us in mind as you think of things you'd like us to help organize and explore. And with that, thanks, Mike, so much for your facilitation today. I'd like to thank Maura McGinty-Kloss, who's been in the background all day with the technology, and have a great day, each and every one of you. Take care, and we'll see you again soon.